Welcome to the MPG Monadnock series primer today. Our talks next week, we have genetic therapies for rare disease, Madeline Oden, and we also have AI in medicine the following week with Mihail Chudich. And today, I'm so excited for this talk, illuminating variant impact with images. And this will be given by two people, so lucky you guys. Anne Carpenter, she earned her Bachelor of Science from Purdue and her PhD from the University of Illinois. She is the Senior Director of the Imaging Platform and an Institute Scientist here at the Broad, where she leads development of open source image analysis tools like Cell Profile and the Cell Painting Assay. She also directs consortium, including the JUMP Cell Painting and OASIS, and is an NIH MIRA investigator, an NSF Career Awardee, and a Royal Microscopical Society Fellow. And joining her today is Runxi Shen. He's a postdoc in the Carpenter Lag, and he earned his PhD in computational biology from Cornell, studying population genetics and evolution. Here at the Broad, he develops computational methods to analyze cell painting data, uncover how genetic variants change cellular states, and also identifies potential therapeutic targets. I'm very excited for you guys to be here. I'm excited to see all these images. Ask questions throughout, please. There's a hot mic right in the middle here. You could just come right up to it. If you just start asking your questions from your chair, the folks on Zoom will not hear. So either flag me down or come to the microphone. And let's go. Thank you.
Thank you so much. Really happy to be here to talk with this group today. I think it's always so exciting to see our technology being used in the space of genomics, and it's kind of a new direction. And just want to let you know the slides are available at the download. I think they are. Maybe we forgot to upload them. Within an hour, the slides will be uploaded at that location, and you'll be able to grab them or take pictures, whatever you like. Everything can be public here. So we're going to start, let me give you a bit of an overview about what we'll talk about. I first want to give some background. This is the primer series, so this is intended to be more tutorial and make sure that everyone's on the same page. So if you know nothing about imaging, you should be in good shape. I'll explain a bit about how images yield single cell profiles, just like single cell mRNA profiles or proteomics. And then we'll talk about 4 different ways that we can use this kind of image-based data. And you can see here looking at gene function, trying to figure out disease mechanisms and do drug screens, and then looking at various variant impacts. And so we'll be talking through that, and Runxi will especially be taking over the coding variant part of this talk. So let's get started. The basic premise of us using images for data is, what if a disease cell could just tell us what's wrong with it? What if there was enough information in the structures and the organelles as they're imaged that could tell us that this certain pathway is disrupted, or this patient has this disease or not? And of course, that must be true at some level, because we know it's true at larger scales. So for example, if you look at the structure of a human face, you can often diagnose a disease straight from the image. And pathologists use this concept all the time, except they zoom in a bit. They might look at your skin and look at a funny looking mole to see if it's cancerous. They might zoom in to extract cells from you and then look at their structure, because fundamentally our biological structures tell us something about biological functions. And there's usually a tight correlation, and often diseases can be revealed in this way. So that's the basic concept that we are taking here, except we are using cells growing in a dish because they're much easier to manipulate. And when we say we're using the cell painting assay, what I mean is we are taking cells either straight from patient samples or cells that have been manipulated in the dish somehow, engineered somehow to reflect a particular disease. We're putting them in culture, and then we're staining them with a number of different dyes that are shown here. And these dyes are every cell biologist's favorite organelles, and they're really informative about what the status of a cell is. It seems like a very simple assay, and that's because it is. It's very inexpensive. The reagent cost is less than $1 per sample. And when we do all the image analysis, it produces a remarkable amount of information. And if you don't quite believe how is that possible, I hope to demonstrate some examples of this today. So the basic kinds of experiments that we do using cell painting involve somehow preparing some samples, and those could be, again, patient cell lines or genetically edited cell lines, or they could be cells that have CRISPR knockdown or an overexpression of a gene or so on. So we prepare some samples that mean something to us in the context of the disease that we're studying. And then we measure everything about those cells. And we use image analysis software, such as Cell Profiler, which was invented by my lab and here at the Broad, maintained for the past 20 years and kept up to date with all kinds of new algorithms that allow us to really accurately identify the nucleus of the cell or the borders of the cell from every individual image. So we measure not just how big is each object, we're not just counting cells, we're doing things like measuring the characteristics of the shape of the cell border, of the nucleus. We're looking at the intensities and textures of all the different stains that we have present, looking at their correlations and relationships as well. So we end up with thousands of features for every individual cell that has been measured in the image. So we can use classical techniques in order to extract those measurements that have some biological meaning to us, area, shape, and texture. We can also use deep learning feature extractors, and I won't go into detail here, but the basic idea is you put in the raw image of the pixels, and you allow the deep learning network to learn what are important features that exist within the cells, maybe beyond the things that biologists think about. So the good news is it can capture a bit more than our set of 1,000 features that we typically measure with Cell Profiler. It can capture a bit more. The downside is those features are not very human interpretable. So we don't have great ways of figuring out what exactly they're measuring. So either way, we end up for every individual cell, we get sort of a row of this data matrix. A typical experiment would have 10,000 times more data than is shown here because we have thousands of single cells for every sample. We might have thousands of samples, and then we have thousands of morphology features. So why do we use imaging and why are we so excited about this as a data source is because it's so much cheaper than mRNA and protein profiling, and yet we find that it has pretty similar predictive success in a number of different tasks that we've attempted over the past decade or so. We find that mRNA and images are typically equally powerful, protein profiles as well. And even more exciting is they're complementary, which means some information you could get using either technique, but there's a lot that you can get. It's usually about a third, that a third you can only get with mRNA, a third you can only get with images. So it's really nice when we have multi-omic data sources together with imaging to really capture a lot of what is going on with a given sample that we're trying to model. Okay, so this is, again, the basic concept of image profiling is we take images, we turn them into numbers, into single cell profiles, and then we do stuff with them. And today's talk is mostly going to be about what do we do with those profiles. We can basically measure, are these two samples similar to each other or different from each other? We can answer questions like which of those features is predictive of some other property that we care about the samples, and so on. Any questions so far? That's the end of that little chunk. Yeah, please.
Sorry. Thank you. Can I ask just a very quick question, actually? Are there ways that are already defined in which ways we can integrate basically the imaging data of a given data set plus the mRNA?
So data integration across modalities is a really fascinating problem. I think it's a little maybe easier to grasp if you're using multiple molecular techniques, but then images are just such a weird data type compared, at least mRNA and epigenomics and proteomics, they map to particular genes, whereas images is just a very different kind of data. So very different equals great because now we have more information, but also more complicated. So there's two ways, there's two classes of ways to integrate data. One is called early fusion and the other is called late fusion. So early fusion would just say, hey, if I've got a certain sample and I have this many morphology features, I'm going to just tack on a bunch of mRNA features and a bunch of proteomic features or epigenomic or whatever else you have, and just put all the raw features in and do whatever task you're trying to do, giving it access to everything kind of equally. And then the other approach is called late fusion, which is analyze each modality independently to make some kinds of predictions and then combine the predictions at the end. And we usually find late fusion works better, but I think that's only because not enough people are working on this space, and there's got to be more clever ways of integrating the data types earlier.
And just out of curiosity, when you say this one third of explaining something about the cell biology, it's using the early fusion or the late fusion, or
that's using each of them independently. Yeah. So if we use them independently, then using both types independently, one or the other will get a third, and then both of them will get a third of the correct answers. And then you're asking, like, what if we actually combine them and integrate them? We so far have not found that integrating the data does much better than treating them both separately. So in other words, late fusion seems to work better.
Very complementary.
Yeah.
Okay, thanks.
Yeah, thanks for those questions. Anything else before we get to the actual, what are we going to do with all these profiles?
One question.
Sure.
Is there a sweet spot comparing the imaging resolution and the imaging speed?
Yeah, for sure. So it's exactly a trade-off between how high resolution are the images and how long is it sitting on the microscope, and that really adds up. It's actually a significant cost of the assay is just renting the microscope per hour if you're at a place where you actually have to pay for it. So what we find, weirdly enough, you would think the higher and higher resolution you go, the better the data quality will be. It turns out there is kind of a sweet spot around 20x objective lens is pretty good. And apparently going further than that, you get a bit more detail about what's going on within the cell. At some point you hit the limit of light microscopy and you're not really getting any more anyways. But I think the real issue is that the more you zoom in on the cells, the fewer cells you capture. And so you have to capture a lot more fields of view to get the same number of cells. And at some point it just sort of deteriorates. So we found 20x to be a really good trade-off.
Thank you. Thank you.
Okay, so let's go to one of the first applications. I want to understand disease mechanisms and make a cell-based functional assay for my disease. Why do you want to make a cell-based assay for your disease? Because then you can do all kinds of things. You can screen drugs. You can also do genetic manipulations. You can try to figure out what's going on with your disease mechanisms. Okay, so what's an example of this? We have this vision of the future where we could find a cell-based phenotype for every disease, and that would be quite delightful. Here's one example where you have cells in a healthy state and then cells in a disease state. In this case, it was a CRISPR knockdown for a loss of function disorder, and you get some kind of visual change. And once that happens, you can then find drugs that reverse that disease phenotype by taking cells that are in the disease state, testing drugs one by one, and seeing, do any of these drugs make the cells look healthy again? That's the basic concept. And so here's that particular example, actually comes from the University of Utah. And the basic idea, they weren't using cell painting, but they were using a few dyes that you can see here. And when they knock down this gene that causes a human genetic disorder, cerebral cavernous malformation, you see a very obvious visual change. And of course, that can be quantified very readily by image analysis. And then they tested a bunch of drugs. And long story short, eventually went to clinical trials and launched this company Recursion, which takes this basic approach of knocking down loss of function disorder genes and then screens drugs to repair the cellular mechanisms that have gone wrong with that disease. So they now have multiple clinical trials running. That's one example. Another example comes from Melina Klausnitzer's lab. I'm sorry that all the credits are covered by the closed captioning here, but from Melina Klausnitzer's lab, they took a different approach of how do we create a disease in a dish. Here, they took patient cells that were at high risk or low risk of developing metabolic disorder. So they weren't necessarily experiencing symptoms currently, but they had the genetics that would indicate that they're going to be susceptible. And that is scored by a polygenic risk score. So you have high and low risk patients, and they just took the tails of each, took patient samples, primary cells from them, adipocytes, and then looked at them in the dish. And what they found is that again, a very obvious difference here. You can see the lipid droplets. They chose to replace one of the cell painting dyes with lipid droplets. And you can see a very clear difference here. Interestingly, though, you don't have to label the lipid droplets directly. There's enough information in the other channels of cell painting to have revealed this disorder. We just didn't know that until we did the experiment. So that's kind of a neat side effect, is that you don't always have to stain for something obvious that you think is going to be disease related. Sometimes the data in the assay just tells you something new that you might not have realized you were looking for. So those are some examples of how we find disease phenotypes so that we could eventually do drug screens and so on. Another thing you can do with images is figure out what a gene does. Yeah, please go ahead.
I have just one more, and actually just because of the previous slide, it makes me think, so I know this is actually combining polygenic information and for one individual, but it makes me think about EQTLs or any sort of QTL where we can actually map the effect of a variant to a given phenotype. And so is there any phenotype that we can actually map to SNPs in that way?
Absolutely, and that'll be the subject of like the next application after this. Exactly, yeah, you're thinking exactly the right direction. Okay, so I know what this gene does. I want to know what this gene does. Here, I'm sorry, I don't have a ton of visuals about it, but I just want to point out that we have collected a ton of data that allows you to answer this question for about 75% of the genome. So if you've got a gene and you don't know its function, 13,000 of them we've overexpressed, 8,000, and you can't do more than that because at some point the genes are too large to express well. So that's kind of the limit on overexpression. We could have done more knockdown if we had more funding, but we did 8,000 there, and then collectively that covers about 75% of the genome, as well as a ton of small molecules. And what we did for each of these, we turn up each gene, turn down each gene, and then take a cell painting set of images and extract the profiles and so on. All this data is now publicly available. This was a large consortium with a number of really great pharma partners who helped to collect the data, as well as, of course, some data production here at the Broad. And this is, you can go to broad.io jump and figure out how to access the entire data set if you're computational and want to do that. But we also have these interactive tools from Alain Fernando Munoz Gonzalez in the group, postdoc. He created some tools that make it very easy to answer, type in your favorite gene and you will get pictures that shows you this is how the cells look when that gene is knocked down or overexpressed. And then it will also tell you what other profiles are the most similar. And that's super valuable because if you have a gene with unknown function and you find that it's highly similar to 3 other genes, you can then go and see what do those 3 other genes do, what are their annotations, and it starts to give you some clues, some hypotheses that you might pursue for your gene of interest. And it can also answer, these tools online can also answer what are the most distinctive morphological features when you knock down your gene or overexpress it. So in any case where you're digging into a gene and putting together a story about it, this might be a useful additional piece of information to add to a paper where you can say, and also the gene is messed up in terms of its mitochondrial shape or its nuclear size or some other parameters. And we're happy, we have office hours routinely at the imaging platform, happy to help you interpret what do those features actually mean if you're using tools like this.
Yeah, quick question.
Yes.
What tissues or cell types are?
Yeah, so this experiment was done in U2OS cells. That's an osteosarcoma cancer cell line because it's easily cultured and grows at densely enough, but also nicely flat enough to get good measurements from. So far we've only done one cell type with this. You can imagine repeating this for a neuronal cell type or hepatocytes and so on. That would be quite lovely, but also expensive. All right, and next we will move on to, I want to know this non-coding variance impact, getting to the QTL question that was asked before. So here, in my vision of Broad's future, this is something we've been talking about for quite a while. Is we started out with a strong history in sequencing genomes, and it's led us to so many variants that now need characterizing. So what comes next after we have variants, non-coding or coding? And we would love to see. There's so many technologies at the Broad, cell painting as well as transcriptomics and proteomics. Can we do a full suite of characterization on tons of variants in parallel so that we end up with being able to, at the very least, classify variants. We might not know what the classes mean or do, but at least if all of your variants for a disease fall into 3 classes, you can pick one from each and then pursue them, for example. But it also provides us with phenotypic insights. Sometimes we get real mechanistic insight from looking at the data of these different kinds of profiles. And eventually this hopefully will lead to therapeutics. Okay, so that's the basic concept. Most people in the room know more about QTLs than I do, but it's a locus that correlates with some quantitative trait. And so those traits historically have been gene expression, the most predominant one, but they could easily be cell morphology. And so we worked together with a whole team that is shown here, Shoma Rechuri and Ralda Name's group. And you can see all the folks involved in this, where we they took iPSCs and plated them, which is really not easy to get a lot of cell lines growing together. So it's very easy to take one cell line and add a lot of perturbations because everything's kind of consistent and so on. But getting 297 cell lines to grow independently, but try to keep them growing at the same pace so that they're all at the same crowdedness when it's time to image them is really quite a challenge. But Matt Tegmeyer was up to this challenge and made this work to the point where we then did cell painting, and there was genome sequencing of these same cell lines. And then we just asked, can we take those morphology features and correspond them to any particular variants? And so again, the citation is right under the captions, but you can download the slides later and see the paper where it goes into great detail about what we found. And in the end, we found that these 297 cell lines were just barely enough to start seeing both rare and common variant associations here. We call them CM-QTLs for cell morphology QTLs. And I think it says MQTL was already used for something else. I forget what it is, but not morphology. So, so we call them CM-QTLs. And then lastly, the last big application, which I'll turn it over to Runxi, is having to do with coding variants. So I'll start with an older story from our lab where we found, we took alleles that were found in lung cancer, so there's somatic genetic variation. We took 353 alleles that were found in lung cancer where it wasn't known which ones are actually causal and actually really doing something and which ones just happen to be passenger mutations in the cancer. We did cell painting and then the image analysis to create these profiles and then did clustering on them. And as you can see, the different clusters in this morphology space have different phenotypes and the cells look different in each one. So in fact, the majority of alleles actually did have a phenotype. They were distinguishable from negative controls. And genes in known pathways actually cluster with each other, which was another exciting outcome from this. And here's just another way of looking at that same data, but just zooming in on one particular gene. So this is just BRAF, and we had a dozen or so alleles here. And it was pretty exciting because you can see the normal version of BRAF. It's down here, the marked wild type down at the bottom. It actually clusters with empty, which is our negative control here. So normally when you overexpress BRAF, the cells you don't notice it by cell painting. There's no particular morphological change. But when you overexpress one of these other guys, these are all some of the nasty driver mutations for cancer. You see a whole cluster of these guys together, but then you also see a separate little cluster, which are known functionally to be not constitutively activating mutations. And so if you were to, it now becomes possible to test every gene, every allele that you could possibly get in BRAF all at once, cluster them like this, and if your mutation in your cancer has never been seen before, it's at least useful to know, does it fall into this category and therefore might be helped by medications that are useful for some of these other guys, or does it fall into that category? So that's the basic concept, but of course, across all the genes and all the variants. And so we really love the idea of scaling this kind of an experiment up to be able to produce catalogs that allow us to more readily predict variant impact. And that leads us over to Runxi. So I'll hand it over now.
One quick clarifying question online. Are there enrichment libraries for genes that have similar profiles after they've been interfered with from CRISPR?
Are there separate libraries?
Are there enrichment libraries for genes that have similar profiles after they've been interfered with via CRISPR?
Oh, well, I'm not sure I know what is meant by enrichment libraries in this context.
I think it was back when you were talking about the JUMP and that there was enrichment libraries where you could look up genes that were behaving similarly.
Yeah, so we have the data for the whole set. And so if you type in your gene name, you will get the rank ordered list of the genes that are most similar to your particular one. So we don't have physical libraries prepared like for each gene, here's its neighbors or whatever, but you can see what those genes are and then, hand cherry pick a library as you like. Yeah. Is there anything else online?
No, that was it. And feel free to clarify that question, Jared, if that wasn't correct.
This is Pam. Can you hear me? I actually have one question about JUMP data. How transferable are the expression or knockout findings from the U2O2 cells to other cell lines? I mean, I guess it depends on the gene, but do main features of the cells change in a similar way in different?
Yeah. I'm glad you asked this question because everybody would be quite right to be a little skeptical, like, oh, what's the point? You knock down and overexpress these genes in a single cell type. But that's really unfortunate because I study neurons or a liver disease or something else. And like, what are the chances this random bone cancer cell line is going to have the right phenotypes? And I guess the reassurance that I have, so of course, sometimes you will not see a phenotype in this cell line, whereas you would if you used a more specific cell type. But as reassurance that it's not hopeless, we actually find that something like, I'm sorry if I don't remember exactly, but something like 30 to 40% of human genes, when knocked down or overexpressed, produce a phenotype in this cell line with the cell painting assay. So 30 to 40% is not bad coverage for a single very inexpensive assay. And we also find that something like 70% of FDA-approved drugs actually produce a phenotype in a single cell line. Those FDA-approved drugs are for all kinds of different disorders. And so we are capturing a broad spectrum of biology that is in many cases conserved from one cell type to another. So we certainly have specific examples of brain diseases where we see a phenotype in a random epithelial cell line. So it is definitely not hopeless. In fact, more often than not, we really do see things in the quote wrong cell type for a given disease. So it's at least worth a shot. It's low-hanging fruit. It's worth a shot. But we would love to also be expanding a lot of these experiments to fancier cell types.
Okay, cool. Well, first, thank you, Dr. Carpenter, for an excellent overview of how we can use the cell painting assay to tackle all different kinds of biological problems in general, and also in the genetic research, which also sets perfect stage to what I'm going to talk about next. But before I start it, I just want to express my thanks to both the organizer today and also my PI for having me here. And also, it's a pleasure to be here today and then present my work at the MPG Primer series. Cool. So going forward from Dr. Carpenter has just talked about regarding how we can use the cell painting assays to decode those coding variants, I'm going to dive into a specific project where instead of focusing on specific variants in a disease or a single gene, we're trying to capture and test the functional impact of thousands of variants at the same time. So this is actually a part of the VarCHAMP initiative funded by the NIH, where we're aiming to characterize the functional impact of more than 30,000 genetic variants across more than 2,000 rare disease genes across a whole human Mendelian proteome. And the idea behind this is because in the genetic research right now, even though we have found so many different associations between the variants and the disease phenotypes, we have limited knowledge about how these variants is impacting the disease in different biological mechanisms. And the idea of our project is to use multiple different kinds of assays to give a comprehensive picture of the functional impact of these variants. So here we're doing a lot of different tasks, like we're doing these protein stability assay to test whether the protein will be degraded or not after mutation. And we're using protein-protein interaction assays to detect how the protein-protein interaction is going to be disturbed after mutation. And of course, we're also going to do a lot of imaging stuff, as I will talk about today. And eventually, the idea is that we use all these different kinds of assays to give a comprehensive picture of the functional impact of different variants, and then combine them with some kind of AI predictor, such as alpha missense, to predict those missense mutations effect, and then use our assays to help us understand the biological mechanisms underlying those predicted variants, and see if we can understand them in a better way. So as you can see here, so far in this project specifically, we have profiled more than 1,000 variants across the whole thing. And if we look at the clinical annotations of all these variants, we can see that more than a quarter of them have no clear clinical annotation regarding their clinical significance, which basically leave all the doctors and the patients who are diagnosed with this variant to be uncertain about their diagnosis. At the same time, even though more than 50% of them are pathogenic, we still have no idea most of them are doing regarding their pathogenicity. So in this case, using our imaging assay, we can detect their phenotype, such as protein mislocalization, which is a very nice representation of their biological function. For instance, in one of our pilot studies conducted by Typoli Lab at the University of Toronto, we find some mutation from the LZTR1 gene, which had a very prominently different phenotypes compared to their reference. And this variant is actually associated with a rare disease called Noonan syndrome, which typically happens in the babies, where it stops the typical development of different human bodies. And after such phenotypes has been identified, we can then use different drug screening to find the drugs that can reverse the phenotype back to their original state. And in this specific project, we actually optimized our assay to capture the protein localization better. Unlike the traditional cell painting assay, where we use 5 different channels to capture the general cell structure, we actually replaced one of the channels with the green fluorescence protein, where we can tag the protein of target, and then using the other 3 channels to capture the general morphology of the cells in general. So in this case, we can use this variant painting assay to capture both the protein localization effect as well as cell morphology to give us a more comprehensive picture of the cell phenotypes after mutation happens. And in our general workflow, both experimentally and computationally in this project, is that first, we're going to overexpress a single allele, either its variant or its reference allele, on the U2OS cells, and then put them into hundreds of different 384 well plates. After that, we're going to stain each cell with different type of stains that we get from the variant painting assay, and then take the microscopic images of each cell in there. And then we will do the computational analysis, where we use Cell Profiler to do this image segmentation to help us identify each single cell in there, and then extract all these different features like Dr. Carpenter has just talked about regarding their counts, shapes, size, textures, and all this stuff. So eventually, this will lead us to about 2,000 features per each cell for our downstream analysis. And with all this informative cell morphology and protein localization features, we then develop an automated machine learning framework to detect if the variant cells have any protein mislocalization effect or alter cell morphology. And the hardest part with this problem is that even though we're able to use such kind of framework to detect whether a variant cell is different from the reference cells, it's very hard for us to tell if it's from simple technological confounding factors, such as wall position effect or some microscopic lining conditions. And to address this issue, we trained this XGBoost classifier to basically train the classifier using the single cell profiles from the same alleles from different control imaging wells, as well as using the different alleles from different wells. And then we're going to use the AOROC metric of this classifier as a surrogate to describe how different the single cell profiles are from different allele pairs or well pairs. In this case, if it's a higher AOROC, it means that our classifier is able to learn the distinguished features between the variant and the reference, which means that there is a more significant effect of protein mislocalization or change in cell morphology. And in order to address the technical variations we observe due to well position effect or the lighting conditions, we actually use the distribution of the AOROC that we obtained from training the classifier on the same well pairs, but from different imaging wells, and then use the 91st percentile of the AOROC as the cutoff for us to call the significant hits of either protein mislocalization and altered cell morphology. And using this method, we're able to identify more than 43% of the genes that we have profiled so far have at least one variant that caused a significant protein mislocalization, which leads to about 15% of variants in total, as well as about 2% of the variants have a significant change in cell morphology. Since the AOROC profile is a continuous metric to describe the degree of difference between the variant and the reference, we can actually see that in the protein mislocalization profiling specifically, all the pathogenic variants are much more likely to cause protein mislocalization than the benign variants, where the conflicting variants and the VUS variants, where we have no idea what their clinical significance exactly, is sitting between the benign and the pathogenic. But our ability to detect the cell morphology right now is not so distinguishable as we can detect the protein mislocalization. And we're still working on this to make it better in the future. And when we're examining the protein structure for when we observe all these variants, we also observe that all the variants that happened within the core of the protein show higher mislocalization than the surface exposed variants. which can be captured by the relative solvent accessibility matrix, where we can see that all the variants buried in the protein core have much higher mislocalization score than the protein that is on the protein surface. And to visualize this in a very nice 3D structure from example protein called factor 9, we can also see that all the variants that are mislocalized are more likely to be contained within the protein core rather than on the protein surface. So after we have identified all these mislocalized variants, the next natural question will be, where do this variant move the protein? So in some cases, the direction of movement is very straightforward. It's basically move the protein from one cellular compartment to the other, like we're showing here using the example of the rhodopsin protein, which the mutations in this protein is very closely related to disease like night blindness. And in this case, when we're using the cell profiler features to analyze the protein co-localization patterns, we can observe that there is a significant increase in protein co-localization between the GFP channel and the mitochondria channel in the cytoplasm. As well as when we're looking at the images, we can also observe the same trend of this gradient of change between the variants corresponds to exactly the feature we observe on the left-hand side, where initially in the reference, we see the protein is localized generally in the plasma membrane, while in the protein where we have the proline to threonine, we have the kind of least mislocalization from the plasma membrane to cytoplasm, while in the last case, in the cysteine to tyrosine, we have the most strongest significant effect of mislocalization into cytoplasm. And even though all the 4 variants that we have shown here has been annotated as pathogenic variants in ClinVar, only one of them, which is the cis to threonine, has been annotated with the function of ER retention in previous literatures. However, with our assay right now, we can identify the exact functional impact of more variants and annotate them with similar functional impact. But however, in a lot of cases, not all the variants impact can be visually seen exactly. So a lot of the patterns that we observe can only be detected by cell profiler features, and using the unbiased quantitative matrix to evaluate how this protein or how this variant changed the mislocalization pattern of a protein. For instance, here, take the example of the CCM2 gene, which is known to be associated with the vascular malfunction in the central nervous system. We have profiled more than 100 variants in this gene and successfully detect the top 7 most significant mislocalized variants of this gene. And even there are only 7 of them, they are forming 3 different types of patterns of mislocalization for this specific gene. In the first cluster, we observe that there is a significant increase of the protein co-localized with the DNA channel and also in the nucleus, which means that the protein variants cause a more localization in the nuclei than the reference. And in the second cluster, we observe the same thing of increased protein expression in the nuclei, but we also observe a significant decrease in the expression in the cytoplasm compared to the reference. Well, in the third cluster, we observe different patterns of specific protein spatial expression at the edge of the cytoplasm in general. When we're looking at visualize everything into cell painting pictures, we can also see that in general, in the CCM2 reference gene, it is localized in the mitochondria and mostly in the cytoplasm. But the first cluster we identified is actually mislocalized this protein from the cytoplasm into the nuclease. While the second cluster, which is mutations happen at the start codon, is having a dramatic effect. Well, instead of moving the protein from the cytoplasm to the nuclei, it only presents the protein in the nuclei, but not the cytoplasm anymore. And in the third cluster, even though visually you cannot tell the exact difference between the images from the variant versus the reference, we can still use the distribution of the subprofiler features to see that the texture of the protein expression in the cytoplasm, as well as the expression of the protein at the edge of cytoplasm, is much higher in the reference than the variant. And the peculiar case with the last VAL53 to the isoleucine example is that this variant is actually a benign variant on the ClinVar, and it had about 6% of population frequency in the Caucasian population. However, in our studies, we find that this variant is actually is going to increase the risk in the central nervous system related CCM2 diseases, which shows that different variant is actually going to have different cell type and also disease type effect that are going to have, even though the functional impact will be the same, the eventual results of this variant in different disease contexts will be very different. And the last one I'm going to show is to show that our cell painting assay is actually going to review more details regarding different patterns of mislocalization change than some kind of assays. And in this specific study, we're collaborating with our collaborator from the University of Washington, where they focus on a specific gene called factor 9 protein, which I also showed the protein structure previously. And in this protein, they use a multiplex assay where they can high throughput capturing the functional impact of the different variants of this protein on the scale of thousands of variants at the same time. And they focus on the protein secretion and the post-translational modification of this protein, because these are the necessary functions for this protein to stop bleeding and also achieve hemostasis in the human bodies. And when we're comparing our imaging results with the protein secretion assay and their post-translational modification assay, we can see that both our mislocalization scores have very strong correlation with either protein secretion and also their post-translational modification assays. Especially, most variants with very low protein secretion also showed a very high mislocalization score. However, we also do see that there is still kind of limitation of our imaging assay where we could not exactly detect the post-translational modification of these two GLU73 variants with normal protein secretion, which also showcase that we also need to have a more comprehensive picture, like we did in the VarCHAMP project, to use different types of assay to characterize the functional impact of different proteins. But even though we have very nice correlation between our score with the protein secretion scores, we still detect two different patterns of protein mislocalization. For example, the one that happened at the signal peptide sequence of the F9 protein, the cis28ARG, is actually having a very different protein mislocalization phenotype than the other detected mislocalized variants, where there is more shift in the protein expression in the nuclei, where all the other variants mostly only shift the protein expression in the cytoplasm, which shows that our assay can review more biological details and higher granularity of the biological information compared to simply a protein secretion assay. And with all that being said, the eventual goal of this project is actually to construct a comprehensive cell map or variant map of different types of variants across all the different genes. And then we can help people curate everything online using our portal to see what are the common features or the differential features among different variants, different genes in different disease contexts, which is going to be public after we have profiled all the 30,000 variants. But still, for now, thanks to the generosity of Dr. Fallow and Leah Starita, our collaborator at the University of Washington, we also published our current 1,000 variants mislocalization score onto their MaveDB database, where you can simply go on there and search for the variants or the gene that of your interest, and then look at the morphological alter morphology score or mislocalization score on the gene of your interest. And feel free to contact us regarding the images of those variants as well. So after we have identified all these variants and their functional impact, like Dr. Carpenter just showed, the next natural steps will be to find the drugs that can reverse those phenotypes. And so far, combined with our current VarCHAMP study, as well as a pilot study, we have profiled more than 40,500 human genetic variants across 250 disease phenotypes. And we're very excited to further extend this initiative by not only finding the other 30,000 new variants, as well as establishing a consortium called VISTA, where we're partnered up with the people from the pharmaceutical industry to see if they can find interesting drug that can be repurposed to just cure this disease or reverse the phenotype directly. And to just show you guys the concept of reversing the disease phenotype using drugs is going to be successful, we're showing this two examples here where successful drugs can actually reverse the disease mislocalization. On the top here, we have the premature aging disease called progeria, where the initial protein expression of the lemon A is kind of like speckly in the cell, But when a mutation happens, it will lose its speckly expression in the cell and makes the nuclei a little bit blabby. But the blockbuster drug, lennafarnib, is actually able to reverse the phenotype and then make it more looking like its original healthy status. The other example at the bottom is the CFTR protein in the cystic fibrosis, where the protein is initially localized in the plasma membrane. It's not in this CF disease. However, the blockbuster drug from the Vertex is actually able to push the protein back into its original plasma membrane localization, and kind of help the patient to have a speed recovery from the disease, and then even extend the lifespan of this cystic fibrosis patient to their 60s right now. And even more, in order to accelerate and also scale up the experiments more efficiently, we're actually adopting a new technology called full image-based profiling, developed by Bliney Lab and JT Neal's lab at the Broad Institute. In this case, instead of doing an array format, where in each imaging wall we only have one specific variant tested, we're actually going to put tens of thousands of variants in a single imaging wall, along with a specific FDA-approved compound. And in each of the single wells, it can express either one of the 400 possible gene alleles that we're going to test in there. And in order to deconvolute the cell morphological change or localization pattern of each single cell, we can use a in situ barcoding to help us using simple microscope to identify which cell carry which type of specific variant, and then use that to help us efficiently detect whether for this drug treatment in this wall, how many variants can be successfully reverted to their original phenotype. And in this case, it will help us speed up the whole process of drug repurposing in a much faster fashion. And in the end, I hope you have enjoyed a full journey of how we can use cell printing assay to tackle different biological problems, as well as its application in the genetic research. But other than we have talked about before, there are still a lot of different kinds of other applications of cell printing, such as we can use them to identify the MOA of different drug compounds, predict the drug toxicity of target effect, side effect, and even use it to stratify patients for more precision medicine. At the same time, there are also a lot of questions that are pending to answer, such as, can we speed up the process in this experiment or computational pipeline using automation, clever biotech, or maybe some AI agent? And can we surpass the human capabilities of understanding those images using deep learning models to help us extract more meaningful biological context or insight for different downstream tasks? And if you are interested in looking into our research a little bit more, you can also refer to all the nice review papers we have published in the recent years, and then talk to us afterwards as well. And you can find our slide deck here on the bottom of the link here. But in the end, our hope is that collectively, we want this kind of technology, as well as all the other kinds of AI innovations in the drug development, can help us break the curve and then cut the cost and the time for drug development, and to develop better drugs to treat more human diseases. And last but not least, we would like to thank all our lab members for all the work they have done so far, and also our collaborators, especially the image analysts in the BETH lab, also in the Broad imaging platform. Without their effort, none of this project will be possible. And also our amazing funding resources as well. Without all these people and all this money, I don't think we can achieve anything here. So with that all being said, we also have our contact info here at the bottom, and also here again, the slide link. Feel free to contact us and look at the slides afterwards, and we're free to take any questions you have. Thank you.
This is Pam online. I have a question. So Noonan syndrome, it's part of a spectrum of neurodevelopmental disorders. So there are several other genes that cause it, and some other the other genes that cause Noonan, as well as like related syndromes. I'm guessing this hasn't been done yet, but have any of these other genes been tested and seen the same to like replicate the findings with LZTR1?
That's a great question. I think we've only looked at that one variant so far. So I love the question, but I don't know the answer.
Yeah.
You showed a couple of examples of cases where something in some gene change leads to some self morphology change, but I'm wondering if you know of any examples where there's a cell morphology change, but at the tissue level, it's not clear what's going on, and so it's like a negative case of using cell painting.
Where there is a change at the cellular level, but it's not seen at the tissue level is
Yeah,
what you're
or
asking?
where it's clear what's going on in the cell at the cell level, but at the tissue level, it becomes unclear what's actually going
No,
on.
we have not done, most of what we've done so far has been really high throughput, like looking at thousands of variants at once. So we have not followed up on very many things at the tissue or organism level. So it's a great question. I think that if you found some, I mean, we're just sort of generating all these hypotheses. I think if you found something that looked interesting, that would be a fantastic question to go and follow it up.
Thank you for your wonderful talk. I just have a basic question. It's out of curiosity. When you do the model training and decide whether there's protein mislocalization comparing the cells with the mutation and the reference cells, have you compared or did you compare the cells have similar genetic backgrounds, say like polygenic score, using this kind of score, to decide whether these two cells, the cell with mutation and the cell with the reference cells have similar genetic background overall, generally speaking, and then do the comparison to decide whether it is the protein mislocalization.
Yeah. So the cells in both cases are exactly the same. They're cultured cells, so they're as identical as cultured cells can be over time. So we're putting separate samples in different wells, and we're overexpressing the protein. These are only coding variants, so we're overexpressing the protein with and without the mutation. So it's not at the endogenous locus, I think is what you're getting at. Yeah. So the genetic background is identical, and it's just the only intervention is add a protein with or without the mutation.
Got it. Thank you.
All right, let's thank our speaker one more time. Speakers.
Thank you.
Good news, you don't have to move. Just refill your coffee and stick around for the seminar. Thank you, everybody. See you next week.